Hello and welcome everyone to this podcast on histone modification complexes. In the past several sessions, we've talked about ways that histones are modified. So if you remember, we have our DNA here. How could you forget? With the histone complexes on it. And the N-terminal tail here. That could be methylated. It could be acetylated. It could be phosphorylated. It could be ubiquitinated. This histone 1 that links the nucleosomes together. We talked about how it could be ADP ribosylated. And then the session right before this, we talked about all the enzymes that could add these groups to it. We talked about the kinases and the corresponding phosphatases that would remove them. We talked about the HATs, the histone acetyltransferases that could add acetyl groups. We talked about the histone deacetylases, the HDACs. We talked about the histone methyltransferases. We talked about the ubiquitin ligases. But what I want to talk today about is the idea that these enzymes don't work alone. The methyltransferases, they work in large complexes. The acetyltransferases, deacetylases, they work in large complexes. And so we're going to really focus on these two here. The others work in complexes too, but we know a little bit more about these two, and so we're going to talk about histone transferases and histone methyltransferases. The first complex I want to talk about is this complex that we've talked about in the past called NURD. NURD. NURD stands for nucleosome remodeling deacetylase. So one of the reasons we look at these complexes as opposed to individual enzymes, for instance, we know that the enzyme deacetylase removes an acetyl group from the histones. But how does it know which locus to find? Because we don't want to deacetylate all the histones, we just want to deacetylate some of the histones. So how does it know which one to go to? And that's why we're looking at the complexes. We can begin with our two histone deacetylases. And remember, the general feature of a deacetylase is that it is going to repress transcription. It's going to turn the gene off. So we have our HDACs here, 1 and 2. They will still have the catalytic ability to remove the acetyl groups from the histones, but they need some help. They can't just work by themselves. So the next one I want to put on here are these proteins called RB AP46, 48. RBAP46 and 48 has a presumed role in complex assembly. Without it, this complex doesn't assemble as well. And I'm going to put the function of HDACs over here, and they deacetylate. Next one I want to talk about is this one down here called MI2 alpha or beta. Either one can bind here. And remember, one of the nice things about these complexes is that they're interchangeable. Maybe alpha has a function in one situation, but beta would have a different function in a different situation. MI2 here, alpha or beta, they have a role in remodeling chromatin. In this case, helping to form that heterochromatin and maintain that heterochromatin state. The next one I'm going to draw here is, and we're going to call this one MBD. And it can be MBD2 or MBD3. The MBD2 has a CpG methylating binding domain. So this brings us to a really important point here, and that is, this is one way that we can specify which genes to turn off. by having this domain here that can bind to these CpG methylated binding domains, it can specifically target the genes that need to be repressed. Interestingly, this MBD3, it has much of the same sequence similarities as this number 2 here, but it's lost its CpG binding, the methylated form, binding ability. But it can bind hydroxymethylated DNA. The next one I'm going to draw here is MTA. And MTA has a protein binding ability with the H3, the histone 3 tail. So this is bringing it in there to a part where it can deacetylate some of those lysines that have been acetylated that we already know are linked to gene activity. Okay, we have one last one to bring in here into this big complex, and it's GATA, G-A-T-A. Now, GATA does a couple things here. It can bind DNA at these repeats of GATAs. It can also bind other proteins. An example of another protein it can bind to is a protein called FOG1. FOG1 plays a role in red blood cell development. And FOG1 is a repressor. So you can see here, by looking at this large complex, that we're bringing in this domain here that can bind DNA. It brings in a repressor, sometimes FOG1, sometimes other repressors. It's associated with the appropriate histones because it has this H3 binding ability. It can specify particular genes that need to be turned off because of these methylated CpGs. It brings in this Mi2 to help remodel the chromatin, maintaining or forming this heterochromatin. And we know that's important because forming heterochromatin, this compressed chromatin state, is important in keeping a gene or a region of the chromosome repressed. And we also then have these histone deacetylases, which we know we need to remove those acetyl groups in order to repress transcription. So altogether, we can see several lines of attack to turn off a gene's expression and to maintain that repressed state, which is important when we think about epigenetics. Because with epigenetics, it's not just turning off the gene for right now, it's turning it off for several cell divisions, and maybe even permanently off. Okay, I want to now move to another deacetylase complex. This other complex is called the SYN3 complex. Like the NERD complex, SYN3 also turns off gene expression. So let's go ahead and draw a piece of DNA here with some histones on it, like so. Let's make it a little bit bigger. SYN3 largely plays a role in bringing this complex together. And so it will bring in a histone deacetylase to deacetylate these histones. It is often found associated with these proteins we've talked about before, MECP2s, which methylate the DNA. And remember, we have then our SYN3 here. Now, it's not unusual to also maybe have one of these proteins here, if you remember, BRM, which is linked to those SWI/SNF complexes. And those are usually involved in turning a gene on, but sometimes they're necessary to open up the DNA just a little bit. And this isn't the whole complex, this is just one of the proteins in that complex. Just to open up the DNA just a little bit in order to get it methylated, in order to have other things happen to this chromatin to put it in its repressed state. SYN3 has a role in proliferation, so it wouldn't be surprising to find out that SYN3 also has a link to cancer, apoptosis as well, and a general sense of maintaining homeostasis. We know it's important because a knockout of SYN3 is embryonic lethal. A conditional knockout, which is simply a knockout that under some growth conditions or under some developmental conditions the gene will not be expressed, but under other conditions it will be expressed. Or sometimes they're just expressed at, say, half the rate, not quite full expression, but knocked down just a little bit. And in these cases here, with a SYN3 conditional knockout, we see developmental defects. Now I want to talk about a specific example with the role that SYN3 has in turning genes off. Some of you may have heard this story before, but others have not. So let me go over it real quick. We know that for the cell to go from G to S phase, I'll finish writing the rest of the cell cycle too here, but we really want to focus on this cell cycle checkpoint here. In order to go from G to S, it has to pass this checkpoint that says there are no mutations and that the cell is ready to move into S phase, all the enzymes are ready, the cell is an appropriate size, all that good stuff. And so I want to talk about a particular checkpoint here. And we may come back to this later on as well. But it's this gene called E2F. E2F is a transcription factor that will turn on genes necessary for this S phase to proceed. So I'm going to just put G1 to S genes here. E2F will bind to this promoter, as well as some other genes, not surprisingly, to turn these genes on, a whole set of genes necessary to proceed. However, if this checkpoint is going to be held in place, E2F cannot bind to the promoter. And in this case, E2F binds to a protein called RB for retinoblastoma. And as long as retinoblastoma protein is holding it, we won't turn these genes on and they will be turned off. The way that E2F is released is that RB is phosphorylated. And once RB is phosphorylated, then E2F can enter into the nucleus and turn on gene expression. There are other ways for this to happen. For instance, if we overexpress E2F, then there will be too much of it in the cell and not all of the RB can bind it all. In that case, E2F will enter the cell and turn on transcription. So where does SYN3 come in? That's what we said we were going to talk about. Well, attached to SYN3 and this HDAC here, we can have another protein. And this other protein is called EBP. EBP is specific to certain genes, and one particular is E2F. So here we have our E2F gene, which is being actively transcribed here. But what EBP will do in this complex, I'm just not going to draw the whole complex, EBP will shut down the expression of E2F. So even in a case where you're overexpressing E2F so that it's not binding to RB and you're turning on these genes going from G1 to S, setting a stage for cancer, if you overexpress EBP, we can shut down the expression of E2F, or at least reduce the expression of E2F. And that's all we need to do. We just need to reduce it so that the stoichiometry is set where E2F is at appropriate proportions where it can bind to retinoblastoma protein. So again, EBP as part of this SYN3 complex can come along here and turn off the expression of E2F. If you turn off the expression of E2F, it won't be entering the nucleus as efficiently and won't be turning on these genes to allow the progression through the cell cycle. Okay, the next thing I want to do is talk about a HAT complex, the histone acetyltransferases, the opposite of these HDACs. So these histone acetyltransferases complexes. HATs are often associated with transcription activation machinery. One we've already talked about, and that was TF2D. Remember, TF2D is the transcription factor complex here that brings in this protein called TBP for a TATA binding protein because it binds to the promoter known as the TATA box. Associated or linked with this transcription factor 2D is something called TAF145. There's a whole bunch of TAF proteins that are associated here. I'm just going to write TAF. I'll talk about some of these in a moment, but this stands for TATA binding protein associated factors. This specific one, this 145, is a HAT, histone acetyltransferase. And then ultimately RNA polymerase 2 comes along here to activate these genes. We talked about this complex earlier in this semester, and it's one that is often talked about in a genetics or cell biology class. I want to talk about Another one that's similar to this. In fact, I'll talk in a moment, it's very similar to it, but not exactly the same. And this complex is going to be called the Saga complex. S-A-G-A. And so we're going to draw this here in the middle, S-A-G-A. There's a few proteins associated with it, even if I'm not showing them all. But I'm going to show a few. All of these TAFs that you see over here are the same. TBP is the same. And we have this other TAF, but it's got a slightly different name, and we're going to call it GCN5. GCN5 is the HAT, is the histone acetyltransferase. All right, I want to say a few things about this GCN5. It's a well-conserved histone acetyltransferase from yeast to humans. It contains a bromodomain. Remember, the bromodomain binds acetylated histones. So it's able to acetylate and then also bind there, presumably to maintain that acetylated state. Within the spectrum of this GCN5s from yeast to humans, there is variability to which lysines are acetylated. In yeast, the following are acetylated: histone 3, lysine 14, lysine 9, and lysine 18. It also acetylates H4, histone 4, lysine 8, and lysine 16. Humans are similar, but they can acetylate some additional lysines, and they also acetylate lysines that are independent of this complex here, particularly as it's linked to TBP. Sometimes in humans, these Saga complexes will exist not associated with TBP. They still turn on gene transcription, but it's independent of TBP. And that's largely linked to this GCN5 variability. So as we see here, we have the Saga complex and the TF2D complex. They look very similar, so I want to list some of their similarities. They both turn on transcription. They activate genes. They both recruit TBP to the promoter. They have some shared TAFs. So they share some of these top binding protein associated factors. The ones they share are 90, 25, 68, 60, and 17. So TAF 90, TAF 25, TAF 68, TAF 60, and TAF 17 are shared by these two complexes. Interestingly, GCN5 and TAF 145 have some redundancy. In some situations, when we reduce or eliminate GCN5 in a cell, TAF 145 can substitute and turn on the genes associated with the Saga. And the reverse is true as well. In some situations, when we remove TAF 145, GCN5 can compensate for its loss. So how are they different? Why do we have both of these if there appears to be this redundancy? Well, it appears that the Saga complex is linked to stress. It will turn on genes necessary to survive a particular stress or to repair DNA once it's been damaged. Sometimes it's thought of as the first line of attack, whereas it'll quickly turn on certain genes necessary to repair any damage caused by a particular stress. In some situations, after it's started transcribing certain genes, TF2D will come along and back it up as well. But we often think of the Saga complex, and if it helps you remember, think of it starting as S, it helps in stressful conditions. TF2D, on the other hand, is less regulated. It turns on genes that are needed for what we might call housekeeping duties, housekeeping genes. These might be genes linked to glycolysis, other metabolic pathways. Typically, TF2D complexes have more TAF proteins associated with them. And though both of these complexes are expressed strongly throughout the genome, and they can turn on many of the exact same genes, of all the transcripts that are made, about 90% are done by TF2D, and only 10% are activated by the Saga complex. Now, it's important to remember, both with these HAT complexes and the HDAC complexes, that one of the reasons we have these large complexes and we bring in these various proteins linked to these complexes is so that we can maintain a piece of chromatin that is actively on or actively off within a cell for multiple generations of cell division. Having this degree of regulation, these many proteins involved, helps maintain that state of repression or activation. Okay, that's all I'm going to say about acetylation or deacetylation for right now. We'll come back to it later. But I want to move on to methylation.